Hi, thank you very much for being here. My name is Jhael Ortega. I am from Ecuador, and now I am a master's student at York University. So today I want to share with you one of my undergrad projects that it's called Systematics of the Genus Noblela with a new genus, 6 new species, and insights into its historic biogeography. So the species that I work with are part of this amazing group of frogs that it's called terrarane because of their terrestrial behavior. So this group is highly diverse and comprises species that are distributed in Central and South America. And so far it is composed by 1,256 species, which represent more or less the 15% of the world's amphibians. So one of the defining traits of terrarana is that they are direct developers, which means that they don't have a larval stage, unlike many other species of amphibians. So this is an adaptation to their terrestrial life and reduces the need for an aquatic environment for reproduction or any other activities, which might have contributed to their massive diversification. So just give me a second, I don't know. So terrarane is composed by 5 families, with Strabomantide being the most diverse one, and within Strabomantide we also have 4 subfamilies. The genus that I work with is part of the subfamily Oloadenine. So Noblela is a genus of small strabomantid frogs that are distributed in mountain forest and lowland rainforest in Bolivia, Ecuador, and Peru. And as currently defined, it is composed by 17 species, and it also includes the species that are among the smallest vertebrates in the neotropics. So taxonomically talking, the history of Noblela has been quite conflicted since its origins. So it was first described by Barbour in 1930, with Smithilus peruvianus being the type species, now Noblela peruviana, and this species was found in southern Peru in a locality that is called Puno. So as I mentioned, the history has been quite conflicted, so Noblela has been synonymized several times with other genera, for example, Phrynoppus or Cycrophrynella, but finally, De la Riva in 2008 resurrected the genus Noblela after reviewing all the type material and realizing that they were in fact a different thing. So despite having a relatively small number of species, Noblela presents some taxonomic problems that are still waiting for resolution. And as you can see in this phylogeny, basically the genus is paraphyletic, which is not great. And this result has been found in many studies, including this one and also this one. But in this one here, Reyes-Boy and collaborators found that this paraphily kind of has geographic correspondence with a northern clade. I'm sorry, I can find the pointer here. With the northern clade distributed just in Ecuador and northern Peru, and a southern clade distributed in southern Peru and Bolivia. So with this in mind, the objectives that we have were to resolve the paraphily of the genus Noblela, identify morphological synapomorphisms of the group, evaluate the species content of Noblela, focusing our efforts in the northern clade, describe the new species that we found, and explore the influence of the Andes in the diversification of Noblela and all its related genera. So we did a lot of things, but I'm going to explain just a few of them now. So I'm going to start with the phylogenetic analysis, and we generated new sequences of these 4 genes for 44 individuals. We edited them, and then we built our matrices, including all the sequences of other Oloadenine species that were available in GenBank. And then we estimated their phylogenetic relationships and their maximum likelihood criterion. We also did a divergence time analysis, and for this we obtained dated phylogeny using the programs BIS2, and we set the divergence times based on the timeline that was proposed by Jhael and collaborators in 2021, and we set only one calibration time at the node where Pristimantis and Celtomantis diverged, which is 54 million years ago. We also did morphological analysis because we wanted to delimit the species, and for this we evaluated qualitative and quantitative traits and some osteological and geometric morphometry analysis, focusing on the shapes of the skulls of the individuals. So for this we obtained high-resolution CT scans, and we set 44 landmarks that you can see in this figure here that were analyzed using the package Geomorph. Finally, we did ancestral trait reconstruction analysis using different set of data. So the first one is the biogeographical information that we had, and we did this because we wanted to assess the effect of the Andes in the diversification of these frogs. So basically here we compare the presence of the species between the Pacific and the Amazonian basins of the Andes in Ecuador. And the other data sets were basically morphological information. So we did this because we wanted to estimate the synapomorphism of the group, and we worked principally with discrete traits. Once more, we did this analysis under a maximum likelihood criterion, and each trait was treated as a binary character under an asymmetric two-parameter Markov K-mode. So here you can see the first part of our results. So this is our concatenated phylogeny that shows that Noblela, as defined by De la Riva in 2008, it's actually paraphyletic. So based on our results, and in order to solve this paraphily, we proposed the following things. First of all, we kept the name Noblela for the southern clade. Because the type of species was found there, and this clade is going to be distributed just in southern Peru and northern Bolivia, and it's closely related to other Peruvian genera. And for the northern clade, we found two interesting things. The first one is that the species that were actually Noblelas of the northern clade, for this species, we resurrected the genus Philonastes, and another group that we thought it was a Noblela, wasn't. So it's the new genus that we describe and that we call Turcufrine, and this is basal and the sister to Philonastes. So they both are distributed in Ecuador and just Philonastes in northern Peru, and they are closely related to Varicolos, which is also distributed in Ecuador. I'm sorry. So here you can see a map of the distribution of the species, and just for you to see, here's the northern clade, here's the southern clade, here's the southern clade, and between them there is a geographic gap that it's kind of 1,300 kilometers in a straight line, and the species in the southern clade also reach higher altitudes. So as I mentioned, we focus our efforts in the study of the northern species. So here's the part of the phylogeny corresponding to that part of our study. So within Philonastes, which is here in green, we found 17 confirmed candidate species, and so far only 8 of them are described. So we included the description of 6 more new species in this manuscript, and with these descriptions, we increased in a 75% the number of species that Noblela has. I'm sorry, Philonastes now. And as part of the review, as I mentioned, we also discovered a new genus of frogs that is basal to Philonastes and sister to it, and it has like really big genetic distances and also some morphological traits that are distinctive. And one of the most interesting ones was the shapes of the skulls. So here you can see the result of our geometric morphometry analysis, and here you can see the specimens belonging to the new genus, and as you can see, it basically has lower values along PC1 and PC2, which means that they have shorter skulls but broader maxillas. And within Philonastes, we couldn't find like clear patterns because the sample was a bit small. And nonetheless, we found that the species belonging to Philonastes cluster in 2 different groups. So the first one that is here basically contains the species that are part of the clade E in the phylogeny. And these ones here basically are grouping species that are part of this clade B. So as I mentioned, we also wanted to estimate the synapomorphism of the group, and we found 4. So the first one is the absence of warming teeth. These are the structures located in the palate of the frogs, and the lack of these structures is the first synapomorphy of Philonastes. We also found that the acuminate fingertips and acuminate toe tips are important for this species. So the picture is not the best one, but the very tip of Philonastes is almost always very acuminate, while in other genera, for example, this one, which is Urcufrine, it's rounded. And the last one is the toe distal expansion. So basically, species belonging to Philonastes have big discs in the fingers and toes, which are expanded, unlike its closest genera. I know that this seems to be not like really important, but it's actually important for the frogs. And finally, I get to the biogeographic analysis results. So here you can see the information belonging to the northern clade. And we found here with number 2, you can see the node at which Urcufrine and Philonastes diverge. And this happens more or less 23 million years ago. We also found, like our analysis support, 5 different colonizations events from the Pacific Basin to the Amazonian one. So you can see that colonizations events with those red arrows, and none in the opposite direction. All of these events occurred in the Miocene, with the newest one, being of one of the new species that we described, Philonastes equatorialis, happening just 4 million years ago. And as you can see here, we have more or less twice the number of species in the Amazonian basin, even though the clade originated in the Pacific one. So this basically is suggesting that there is a highest diversification rate in the eastern slopes of the Andes in Ecuador. So going quickly through the conclusions, Novlela is a paraphyletic genus, and we recommend the separation into 2 genera. So we keep the name Novlela to the southern clade, and we resurrected the name Philonastes for the northern one. The eastern and western slopes of Andes still harbor species of Philonastes where most species are unknown, and we found 17 confirmed candidate species within the genus Philonastes, and with the description of the 6 new species, we increase its richness in 75%. Yeah, thank you very much.
Basically, I have recently become interested in studying this particular invasive species, Vespa velutina. And it sounds good, both musical and poetic. The reason that I'm interested in invasive species is basically, is mentioned because of these 3 interesting consequences associated with an invasive species. The first is that it often causes ecological and economic. It has this ecological and economic consequence, and it can impact the local fauna and flora. And it can also cause problems with human health. And this is one typical consequence associated with invasive species. And the other is the population consequence. It is associated with this founder effect. So it often allows the species to cross the so-called fitness valley and ascend a different adaptive peak. And there is also this intercolony selection, particularly associated with this particular life history of these bees, because typically a single queen can often serve as a founder. However, the genetic variation carried by the queen is limited. So you typically would wonder how could they overcome the potential effect of inbreeding. And the third consequence is this biogeographic consequence, because it often not only increase the biogeography, increasing the species diversity by one species, this invasive species, but also causes the loss again of other species. Okay, so because of these, this is of course, often we are curious about why species X is present in species A but not B, and typically we have these 2 hypotheses. One is that species X has never get a chance to go to species B, and the other is that the habitat in species B cannot support the survival and reproduction of species X. But when there is invasive species, then you immediately know that the habitat is appropriate for these species. Okay, and so this has a lot of, so this is a natural transplant experiment, so that allows us to answer some questions. So with these, I will introduce you to this. How come it does not move? Okay, so these species, it is typically in, the center of speciation is in this tropical region. And of course, insects are typically affected by the repeated glaciation. So they get pushed to the south, and when the glaciation retreats, they gradually spread to the north. And so these particular species, it is a typical Asian species. However, there are some rows on top that is not displaced. There is France and Belgium that are recorded quite early. So this is Belgium, this is France. Then when you have an invasive species, when you come to a new region, the frequency naturally is low, so it is equivalent to a new mutation in a population. It doesn't matter if it is advantageous or deleterious, it has a very high chance of going extinct. So this was recorded so many years ago. Then after that, there has been many years without any any sighting. Then again in 1980, again there is a row above. So there are quite a number of species that are observed in a number of individuals observed in Netherlands. And again, after that, it is not observed until in 2005, it is observed in France. Then suddenly, it just spread like wildfire. So at that time, when it was observed, it simply immediately found almost everywhere. And you can see that it expanded very fast. So this is from 1910 to 1914, and this is from 2015 to 2019, and this is, so basically it just spread very fast. Of course, at the same time, it also spread in the Asian region as well. So this naturally would lead you to think, why the species is successful, or in general, what characters are required to make a species a successful invasive species. So generally, you know that the species, they require these type of things. First, they have to disperse to the new habitat, and they have to find food, and they have to have mate and nest, and they have to grow rapidly so that they will be less affected by the stochastic effect. And they also have to purge off deleterious recessive alleles so that they will minimize the inbreeding depression. And of course, they also have to neutralize the new pathogens and dodge new predators, etc. It'd be nice if somebody could think of an acronym to describe all of these requirements. Now, the disperse is often for these, they often attach, the queen often find itself in cargoes, so they can get transported over long distance. So here, and also I will mention these dispersed and these food. So this is another example. I used to have a student who was working in the Canada Border Services, and border services people, they keep illegal immigrants outside or monitoring illegal immigrants, regardless of whether it is human or species. So this is an illegal immigrant. It has been observed in Vancouver and Washington State. It is the giant. This is the giant Asian hornet, and this hornet, especially the one that is the focus of this lecture, this talk, is a specialized predator. They are and of course they cannot by themselves migrate across the Pacific, but they can hitchhike from Asia to North America. So you can trace their source population to South Korea and Japan. The Canadian one is from Japan, and the Washington State one is from South Korea population. So this shows that the migration, the dispersal, long distance dispersal is no longer a problem for invasive species. And the next one is the food. And the food for these species, especially for Vespa velutina, the Asian hornet, and it is the honeybee. It is a fairly specialized predator on honeybee. There is a story about the hornet and the honeybee co-speciation or co-evolution, but here I just show you the geographic distribution of these honeybee this area. And again, the center is in the speciation center is in the tropics and spread to other places. The European honeybee, Apis mellifera, it is also actually originated from Asia. So it migrated to Europe and become diverged into a number of subspecies. Okay. So it is a long story. I will leave it aside. So now come back to this Asian hornet, and this is the phylogenetic tree, and you can see that these are tropical species that are the sister species, and so this is this cluster is the hornet, and you can see that there is a cluster of these Malaysia, Indonesia cluster, and there is the other cluster. And this cluster, it is part of them is in India and Pakistan. Okay, so that is, and the other is the Asia. So it spread more to the east. Okay, and so this is the Japan-Korea clade. There is no genetic differentiation. So all the samples in the mitochondrial DNA, they're all identical. So must be recent invasion into Japan and South Korea. And this is European clade. It is also genetically identical, so it cannot be ancient migration or colonization. So it is just very recent colonization. So there is also this Thailand, and also this European. It is genetically it is very closely associated with these sample specimens from China. Okay. Now. This does not seem to go. Okay, so this is just the clade for the species alone. I think it goes by itself. So this is what is called a geophylogeny. So it is aimed to summarize both genetic variation over time and space. The vertical line is proportional to branch length. So you can see that this is long branch from this Malaysia, Indonesia, and then there is also another clade in India, and then it is spread to east. And if you do not see a vertical line, then it is all genetically identical. So all these, it is genetically identical to these, and all these blue lines without a vertical line, they are all genetically identical. Okay. So that it shows that there is not multiple colonization. It is different from like in Hawaii, for example, in Hawaii Island, for some species, you can see multiple colonization from the Asian continent, but here there is just one single recent colonization. Okay. So this is just a different geophylogeny and just depicted in a different way, simply because when you have use different background, the map, then often you require change the color. So these are all generated from the PGT. Okay. Oh, this is interpretation of what I say. Interesting. So of course, I'm writing this software for displaying this geophylogeny. So if you have any good ideas about how to improve the visualization, how to summarize summarize the genetic variation over time and space in better way, please contact me. Okay. So I have mentioned these prerequisites for a successful invasive. So I also want to mention this growth. So the growth is, you can estimate the growth rate. So this is from France. So you can see that this is the observations, and at this point suddenly become observed everywhere in France and neighboring countries. So they begin to have a dramatic coordinated effort to control it. So from this one, it is more natural. So I plotted just this point over here. So this is somehow a sigmoid curve. You can estimate the instantaneous rate, and you can see that it should started in about 2002, and so the instantaneous rate is very high. Okay. So here, last, I also want to mention how these species can purge deleterious or recessive. So you can imagine. So this is just a hypothesis. So a species. So first, one generation generates some variation with queens. So some queens will become extinct, and the remaining queen will generate new ones. And again, many intercolony selection and leave only a few available. So this continues, And so the result is that overall you get a lot of, you get long internal branches, but shallow terminal branches. And these turn out to be quite consistent with hornet species. So you can see that in other words, there is a prediction about phylogenetic tree shape. You expect a longer one. Of course, there's no hypothesis based on various evolutionary processes, and you expect that this would be longer than expected, and this would be shorter than expected. Okay, so this is basically the conclusion. So you have so it is originally in tropical region and diverged in various places, and this one particular species, Nigrothorax, become very widely spread, not only in Asia, but also spread to Europe. So those spread to Europe, they saw these subspecies. Okay, so you can see that in this clade you can find that some species are very sedentary, some species or subspecies are very migratory, and it'd be interesting to see to explore the population consequences of these different traits. Okay, now the existing observation also suggests multiple dispersals, but the most recent one is actually just started in 2002, okay, by a lineage with a very high intrinsic rate of growth. Okay, so the colony structure of the hornet and this inter-colony selection probably will facilitate elimination of deleterious recessive alleles, simply because you can imagine that many queens go to different places, and if it carries a lot of deleterious recessive alleles, then it is going to be eliminated. But they are through this recombination. Some queens will carry relatively less deleterious recessives, and they will be more successful and will replace those colonies with more deleterious recessives. And this inter-colony selection will lead to colonies with less deleterious recessives. Okay, so that is our finish here.
All right, good morning, everyone. My name is Leland Graber. I am a PhD student at Cornell University in Corey Moreau's group. And today I'm going to talk about the biogeographic history of the harvester ant genus Pogonomyrmex. So historical biogeography helps us understand modern-day biodiversity. It's a framework that we can look at to see how biodiversity evolved and spread across the globe. It helps us answer a variety of different questions, ask and address these questions, namely, what processes, biotic and abiotic, led to current species ranges? Why are some species, like rats, widespread, while others are restricted to small areas, like pandas? And then how did adaptations allow for species to expand their ranges? Like an example is tetrapods getting the ability to walk on land and being able to colonize every terrestrial area, basically, in the entire world. So I feel very fortunate to be working on biogeography questions, namely because I work with a great system to do it. I work with ants, which are found on every continent except Antarctica, which is a little ironic because Antarctica is the only continent that has ant in the name. But ants are a great system to be working on this because ants are very widespread, very ecologically successful, and have a whole range of adaptations to exist in a variety of different habitats. All sorts of different habitats. Now, the setting for today's story is one particular habitat, the desert. And the protagonist of our story is the ant genus Pogonomyrmex, a desert-adapted harvester ant. The genus Pogonomyrmex has 95 valid species. It is known for its seed harvesting and seed-feeding behavior. They eat a lot of seeds. It's one of 3 genera in the Pogonomyrmecini tribe, along with Hylomerma and Patagonomyrmex. It's pretty different from both of these genera, and it's also a lot bigger than both of these genera. There's a lot more Pogonomyrmex species. And Pogonomyrmex is only found in the Western Hemisphere. Its species range kind of breaks down a little bit like this. The greatest Pogonomyrmex species diversity is in these North American deserts. If you've ever been to the area, they're super conspicuous. you're walking around and you see a bunch of them, and you can see up to 15 species like co-occurring in the same place. So a ton of diversity, a kind of species co-occurrence. And in North America, they all kind of exist in very similar habitats, namely dry deserts, dry grasslands, and then in Florida, some like coastal plains, which do tend to be sandy and dry. But there's a lot of the same kind of habitats that they are inhabiting. South America, on the other hand, actually has more Pogonomyrmex species, nearly twice as many as North America. But there's a lot more diversity in terms of where Pogonomyrmex lives, and a lot less species co-occurrence. There's a lot of co-occurrence specifically in these deserts within Argentina, and then some grasslands in like the eastern part of South America. But there's a lot more widespread species. South America is also different because it has Pogonomyrmex that live in really not desert areas. There's Pogonomyrmex that live in humid rainforests and in places where the genus is really not typically found. These groups also tend to be pretty geographically restricted, so in like small pockets that you can only find these species, and they're also usually not very commonly found at all. Some of these we only know from a couple of observations. And then the island of Hispaniola actually has a small group of Pogonomyrmex species as well. We don't know a ton about these either, but they do tend to inhabit more dry habitats. So one of my questions broadly for my thesis work was, how did Pogonomyrmex take over deserts all across the Western Hemisphere? And then more specifically for this talk, where and when did Pogonomyrmex originate? How many times did Pogonomyrmex disperse to other continents or land masses? And then what geologic processes contributed to Pogonomyrmex dispersal and vicariance? There's not a ton in the literature. It's mostly speculation. Nobody's done a true ancestral range reconstruction for Pogonomyrmex that has been published. But there is some speculation about the origins of Pogonomyrmex, and the literature is mixed. So both North American and South American origins have been proposed for the genus. So to set out to test these questions and to test these other hypotheses, I decided to infer a new phylogeny for Pogonomyrmex using ultra-conserved elements, and then infer the ancestral ranges of the ancestors of modern-day Pogonomyrmex. So here are my methods. I'm not going to spend too much time talking about these, especially my wet lab and phylogenetic inference parts, but you are welcome to ask me questions about them after the talk. I do want to mention for my divergence dating, which was necessarily for the ancestral range reconstruction, I used BEAST2, and I calibrated my root age with the age of Pogonomyrmecini from the Myrmecini phylogeny by Ward et al. In 2016. I used this specifically because Pogonomyrmex only has one fossil, and this phylogeny was calibrated with many more, so I feel more confident about its age. And then I did my ancestral range reconstruction using Biogeography using the DEC model. For my ancestral range reconstruction, I used 6 biogeographic regions. These are based primarily on Pogonomyrmex occurrence, and then also kind of biome similarity between these regions. I do want to point out the last region, Brazil and Guyana. It says 1-2 species, but it's actually really just one very unusually widespread Pogonomyrmex. I found it hard to kind of incorporate that species in here, so I gave it its own region, basically. I think it makes a lot more sense with the analysis. And so the results of my work, I'm not talking too much about systematics here, but to give you some context, I got 3 major Pogonomyrmex clades, each of which it looks like it's more than 20 million years old. I have the Pogonomyrmex myri clade at the top there. You can see there's green. That seems like a very early diverging clade of Pogonomyrmex. The Aphibomyrmex clade, Pogonomyrmex, actually used to be 2 genera, one of which Aphibomyrmex comes out as being monophyletic. And then the very large Pogonomyrmex clade, which is the majority of the genus. And now I will take you through several milestones in Pogonomyrmex history, as shown here by the tree. So about 54 million years ago, it appears the last common ancestor of the whole Pogonomyrmecini tribe was living in northern South America. I think this result makes a lot of sense, especially because both of the other genera in the tribe are only found in South America. Although interestingly, my results say that by 46.4 million years ago, the last common ancestor of all Pogonomyrmex, the genus, is likely living in North America. I think this result is kind of interesting, and I treat it with some skepticism, especially because the result of it being from northern South America is almost nearly just as likely as this. So this is one of the areas here in my analysis that I think I'm going to give a lot of attention to in the future. So I would say the last common ancestor of all Pogonomyrmex is somewhat likely living in North America, or some greater region there. But one thing for sure that we know is by 34 million years ago, there was Pogonomyrmex in North America, specifically in Colorado, because we have fossil evidence of it. And then so for the next 20 million years or so, it appears that Pogonomyrmex was in North America diversifying until 14 million years ago, a single Pogonomyrmex lineage returned to South America, ending up in the southwestern part of the continent. That's a pretty big jump. I think that's a big mystery how that happened, but I'm very intrigued, and it seems very likely that it did. One thing that could have happened is the fact that at the time the Isthmus of Panama was forming. It wasn't completely formed, but these chain of volcanic islands might have allowed Pogonomyrmex queens to disperse pretty easily at this point and then end up in that area. And then more recently, lineages within the Ephebo-Myrmex groups were introduced to Hispaniola by 5.5 million years ago and had two separate introductions to eastern South America between 5.5 and 3 million years ago. I was pretty surprised by how recent these were, actually. I wasn't expecting that as much. And I do think it is very likely that the introductions back into South America were much facilitated by the Isthmus of Panama, because we also know that ants that were flightless, that do not have queens and males that disperse via flight, were able to disperse from South America to Central America at this time. So I'm not surprised to see Pogonomyrmex distribution at this point. So to summarize my results, Pogonomyrmecidae, the greater tribe, most likely originated in northern South America, a result that's pretty consistent with what the literature was saying overall. The genus Pogonomyrmex is about 46.4 million years old. Early in its history, Pogonomyrmex migrated to North America, although it's unclear which at point in the genus history it was. And Pogonomyrmex most likely returned to South America 3 separate times between 14 and 3 million years ago and dispersed once to Hispaniola 5.5 million years ago. And I think the next focus of my research probably is going to be looking at how that dispersal to Hispaniola happened and looking more at the details of that. I just wanted to add in here too, one of the hypotheses about Pogonomyrmex origins and dispersal by Fernandez and Palacio in 1997. They go into much greater detail about how they think Pogonomyrmex dispersed and diversified within the Americas. And taken out of the context of this phylogeny, I don't think there's really a lot of basis for it, but reading it again now, after doing this analysis, I think it makes a lot of sense. So Fernandez and Palacio wrote that the ancestral forms of Pogonomyrmex were likely cryptic species inhabiting rainforests and probably having a carnivorous diet. They go on to say that the Pogonomyrmex ancestors were once widely distributed in Mesoamerica and living in human rainforests. And they think that different forces, climatic forces, moved Pogonomyrmex ancestors north, and then they became adapted to dry climates. Some of these adaptations might include developing bigger and wider heads to have an easier time relying on seeds as a food source. So it's just me plugging another chapter of my thesis that is in prep. But it is an adaptation that we found specifically in the Pogonomyrmex group. But one thing is for sure, these adaptations to dry conditions allowed Pogonomyrmex to thrive in deserts and grasslands all across the Western Hemisphere, and that's how they came to be today. And with that, I'd like to acknowledge my lab. I'd like to acknowledge some collaborators, Jacob Landis and Mark Buckner, for help with analyses, my funding sources, and then I can take any questions. Thank you.